Parliament of Israel, and I would like to welcome you to the State of Israel on behalf of a former, very important minister in Israel, but he was the time that he will arrive. Fortunately, when my ancestors had come to Jerusalem, they found here people who came from Yemen, only because the rabbi ordered them to go to Yemen. People from Morocco were here because they were told that in the year 1809, Messiah has to return back to Jerusalem. In 1856 already, the majority of the Jerusalemites were Jews here in Jerusalem because everyone returned to his homeland. And according to the idea of Zionism as a political movement, the word Zionism is being taken from the word Zion. And Zion is a synonym of Jerusalem. And wish you a nice day in Israel. God bless all of you. Thank you.
Thank you, Mr. Speaker. As you see, everything is relative. The smallest country with 7 million citizens. We have 7.5 million. We have so many problems compared to it's like a small street in Beijing.
The honor to join in the special session of the Knesset Science Technology Committee for annual National Science Day, particularly when celebration of Israeli Academy of Science community for 50-year anniversary. I'm also appreciated to be invited by our host to deliver a very few words here titled Our Responsibilities. Environmental pollution and climate change, natural or human-induced disasters, new epidemic diseases and health care for most people, hunger, food and water security, aging population, as well as we have to be secure against the traditional and non-traditional dangers. Anyway, all issues cannot be responded and solved just by one country or one nation alone. Globalization has built a strong interdependent linkage among us anyway.
And we also knew that ribosomes create these proteins according to the genetic code. So just I allow myself to use a book, a children's book called Le Biokit, or in Hebrew Biokit, and they describe some of the proteins that are doing cellular work, like insulin, that is controlling the sugar, the sugar level. I talked about earlier, I mentioned quickly the immune response, collagen, which is the cellular protein, human skin, hemoglobin that transfers oxygen, and trypsin that digest food. All these are proteins. There are 20 types of them. They all have the same backbone, but different additions. And some of these additions can be those that give the proteins the possibility to do their jobs, like tools. So then they come together. This is what the ribosome does. Bring them together, very long chains, much, much longer than those. Here is a chain representing what you see here. And before they fold correctly, they can do nothing. But when they are correctly folded, like here, they create a site called the active site, and that's where they do what they have to do. Active site has to be accurate, fully accurate, and the folding that creates the active site is dictated by the sequence of the amino acids, the building blocks that are making the protein. The sequence is dictated by the DNA. And when I was a student, DNA was just discussed, but now every child knows what is DNA, so I don't really have to tell you what it is. But from our point of view, this is the storage of the information that tells later how proteins will be done, and the information is stored in these four different colors, four different components of DNA that are called bases. So earlier we talked about 20 amino acids. Now we talk about a language that tells the amino acid sequence by 4 letters. So there are here 2 different languages, a 4-letter language and a 20-letter language. And the ribosome understands both of them. It's actually a factory that can read, you see here, the instructions in a 4-letter manner that can attract the amino acids that are coming by carriers. They are drawn here like very, very similar, although not identical tracks. The whole thing happens here together in this factory that needs energy, that when it finishes, it can release the language, the instructions, the DNA type language, the genetic language for being translated again and again, and the protein as a long chain is coming out here. Now the tracks can go and look for more jobs to do. Working all the time together. Even bacterial cells contain a lot of ribosomes. When bacteria grow, when the cells grow, they can reach 80,000, even 100,000 sometimes. And the ribosomes work continuously with hardly any mistake, the most important role in life. Because of it, many antibiotics target them, target the ribosomes. It means when one bacteria wants to get rid of another bacteria, it finds immunization against the ribosomes of the other bacteria. Not all the antibiotics are those, but many of them. Over 40% of the useful antibiotics are doing it. So natural antibiotics are the weapons that bacteria are using when we are trying to eliminate other type of bacteria. And we take advantage of them because they can target bacteria and not the patients that we would like to recover. You can see it now better. Here you look into the tunnel, that's the ribosome, the green thing, and erythromycin blocks it. So the protein is being made but cannot live. And that's very important for the life of the bacteria. See, that means no life. And I want to invite all those that didn't go to the Dead Sea yet to go there because it's a wonderful place. But no life is really no life in terms of fish and plants and so on. Still there are 2 bacteria in one algae, and I want to show you one of the bacteria. Grows here, you see a colony. All the salt that comes out from this very, very salty water. You can even see it better here. And this is the distance of the water that went down from the winter to the summer when this picture was taken. So if there is a bacteria that goes there under such high salt, and sometimes very high temperature, 60 degrees, 55 degrees in the summer, it must have very strong ribosomes. And indeed, using it, we got lots of nice crystals. Not using only that, actually, we are using also thermophiles and halophiles, which are from the Dead Sea, and radiophiles, all kinds of bacteria that are robust. And we got nice crystals, and we could measure them. When we started measuring them. So this is a synchrotron, an accelerator in Grenoble, in France, where Israel is a member, and where I came back yesterday for